With a rich academic and research background, Dr. Giri has previously served in esteemed roles such as Senior Postdoctoral Researcher at IIT Guwahati, Postdoctoral Fellow at Banaras Hindu University, and Postdoctoral Researcher at the University of Hawaii at Manoa. Additionally, he has held position as a Senior Researcher at CSIR National Environment Engineering Research Institute and RTM Nagpur University. Dr. Giri holds a master's and PhD in microbiology from Barkatullah University, Bhopal, and CSIR NIRI, Nagpur. He has completed postdoctoral research at prestigious institutions including the University of Hawaii, Sejong University, and Hanyang University in South Korea. His experience spans diverse fields such as atmospheric science, biochar, bioenergy, anaerobic digestion, and environmental microbiology. A prolific researcher, Dr. Giri has published over 100 research and review papers in renowned journals such as Bioresource Technology, Renewable and Sustainable Energy Reviews, Science of the Total Environment, and Journal of Cleaner Production. His work has earned him global recognition, including being listed among the top 2% of researchers worldwide by Elsevier and Stanford University. Dr. Giri research impact is reflected in his exceptional citation matrix with over 7,562 citations, an H index of 44 and an I-10 index of 84 as per Google Scholar. His contribution to environmental safety are further complemented by certification in industrial safety and lab safety training from institutions such as AIIMS, Chennai, and the University of Hawaii. With over 2 decades of experience, Dr. Giri has worked in leading research organizations, including multiple CSIR labs across India and top-tier technical institutions like IIT BHU and IIT Guwahati. At UPES, he is recognized among the top 10 faculty members and supervises 3 PhD scholars. His global collaborations span countries like USA, Israel, the Netherlands, South Korea, China, and Taiwan. Underlying his international research influence and commitment to advancing sustainability and environmental science. I welcome you, sir, on the behalf of Civil Engineering Department. Kindly proceed for a lecture.
Okay, thank you, ma'am, for a nice introduction, and thanks to Jogani madam for inviting me. So I'm sharing my slides.
Now visible, sir.
Okay, so as we know nowadays, people are like talking about sustainability in a huge manner. So, and this theme is also the we are focusing on the sustainable things. So, as madam told me that I have like experience in this sustainability things. So when I was in University of Hawaii at Manoa, I have started to using the biomass, which are in ample amount in everywhere, even in our country. Like people are telling always that we have a problems when the crop cutting. So there is a parali as a waste. So people are telling that farmers they burn the parali and they are creating the pollution to destroy the environment or somehow making some problems. So taking this account, I suggest that this is not a problem. This can be this parali we can take as a resource to get the like some biomaterials like biochar. We can we can make some like different temperature conditions, and we can make the activated carbon so that they can filter the water and they can filter the like wastewater or waste air, anything. I have done many things. Like I have treated the dye containing wastewater, pesticide containing wastewater using these biochars. And after the treatment, these biochars will work as a biofertilizer. So my presentation will be in the two form. Like one is the like bioresource utilization using the like various aspect, and one I will go through the startup culture in India, like how the startup will help to the our students to get career in these things. So using this thing, like I use that bioresource utilization and use this bioresource to make the mushroom cultivation, and we sell the different mushroom to the society. So let's start with the first slide. So here we have used so see how the biochar form. So generally what we took biochar is a product of the pyrolysis. If our students know like what is pyrolysis, so pyrolysis is a process where we use one pyrolyzer, where we give the uh, nitrogen to make the inert conditions, and various kind of temperature we fix around up 300 to onwards we use. And we need some bioresource, yeah, any waste. Like see, we can see here we have animal waste, like animal dung, food waste, sewage sludge as a waste, industrial wastage, like any pharmaceutical industry. When we go, we found the huge amount of industrial waste. Accordingly, like fertilizer company, we have various kind of waste. And one is the when we go to the like any wood industry where they make the uh, chair, table. So they have the very huge amount of wood chip as a waste. And then another kind of waste is agricultural waste. As we know, our country is the agricultural uh, importance country, and we have a huge amount of agricultural waste after the cutting of the crop. The leftover is we consider as a waste. But these are not the waste. And another one is the like forest waste. When we go in the forest, like I am working in Dehradun, and it is a Uttarakhand region, and maybe students sometime having a news like there is a forest fire. So what happened in Dehradun region or in Himalayan region, there is a pine tree, and these pine tree having a if students know that the pine has a very strong ability to catch the fire very immediately. So when the pine leaves, pine needles get down from the tree, it catches the fire, and it make the forest fire, which is very dangerous for any forest. And these forest fire destroy the uh, human destroy the flora and fauna of this forest, where they where many living being burned in the fire. So that's why we have like a plant key. We will not allow to get the forest fire. Before to getting the forest fire, we will collect this forest waste, and we can fix in the some pyrolyzer, and then we can make the biochar as a product. So now these are the like bio waste. When we have several other kinds of bio waste, and the process will be the pyrolyzer. So we fix these this biomass in the pyrolyzer unit, and we kept the conditions like 300 degree and beyond that, as per our product requirements. And after this pyrolysis, we have the product like bio oil, biochar, and biogas. So when we vary the temperature, so biochar we get more or less, and varying when we varying the temperature, so biochar has a different kind of surface structure, and it has like a structural changes. There have several kinds of pores where they absorb and adsorb the various kinds of pollutant. And another one is the bio oil. Suppose we have some pharmaceutical important plants, like when we go to the agricultural waste, and some plant which have the medically important, and when we make them, so it has several kinds of pharmaceutically important components, and we can find here, and we can use them to treat the any kind of disease. Like I will show the how the bio oil from the arjun seeds they cure the some bacterial killing. I will show in the next slides. And one is the biogas. Biogas is also it found in a very few amount, and these all the products are depends on the which kind of feedstock we have taken. So here is the whole process I shown. Like see, you can see here the like these are the some biomass. It is a Chinese fruit plants, and we washed here and we keep in the pyrolysis chamber where you are able to see that we are purging the nitrogen gas inside to make the inert condition. Students, please remember, in absence of oxygen, the process will happen. It is called pyrolysis. If you give the oxygen, then it will be the burning process. It will not call the pyrolysis. So it is a very important to know. If anyone asks like what is the difference between burning and pyrolysis, so just simply you say this whole process will conduct in absence of oxygen. So that's why we give the nitrogen to purge these inside things to make the inert condition. And then we have the biochar, and then here we have some distillation process, and we can get the bio oil. This bio oil gets here I told you like as per the biomass, we can use them as a bacterial treatment. We can kill the bacteria. And here I am showing the application of this biochar for the dye removal. You can see when I was in IIT BHU, I was working on the like malachite green dye, and we used this biochar. And see in the bottom portion, we are able to see there is a completely absorbed by this biochar, and this the bottom water is almost clear. And we have done this study using some antimicrobial resistance using the bio oil, and the bacteria name were is E. coli, Serratia species, Bacillus subtilis, and Pseudomonas fluorescens. So these bacteria we have used to check the bio oil obtained from the Chinese fruit plants, and we check using the experiment. And here is the biogas. Biogas was obtained in a very few quantities. Just we have like analyzed what content is there. So after this, we have published this paper after doing some basic study, and you found that how the biochars are help to get the agrochemicals and synthetic dyes from the environmental samples. So this paper was published in the Chemosphere, and it was very important paper, and within the 2 to 3 years, it got the highly cited papers. So in this paper, what we have studied, so we have find some agricultural waste. If students know like what is the agrochemical, so agrochemicals are the fertilizers, pesticides, insecticides, and the synthetic organic dyes like malachite green and the things. So you know what happened when the farmers or anyone use the agrochemicals like biofertilizer or pesticide in the crop. So what happened? They don't know the exact health of the soil, and they give a huge amount of these chemicals to the crop, to the land, to get the more product. So suppose if our 1 kg soil need 10 gram of the fertilizer or any pesticide, but because of the unknowing, because of the unawareness things, farmers or anyone give the huge amount of these chemicals, and these chemicals get accumulated in the soil, and they destroy and they disturb the soil health in the environment. So what happens the next, if these chemicals are a very huge amount in the soil, so they kill the earthworm, they kill the so many important insect which help to get the pollinated in the fields, and these are accumulated in the plants, and these chemicals accumulated in the plants, and when the animal eat them as a fodder, and after animals we get some milk. Yeah, some non-vegetarian people eat some goat or any, so they get these chemicals to their body, and this is called some biotransformation of these chemicals, and which make very dangerous effect. So that's why we have to check our soil health, and accordingly we should go to the some soil scientist, and according to their suggestion, they have to use the agrochemicals including agrochemicals which contains fertilizer or any pesticide or insecticide to the soil. So these were and this paper was very important. After that, uh, so this is some process and their application, like how it happened. So you can see like here is the waste biomass, different kind of biomass. We have used temperature up to 700 degrees centigrade in absence of low oxygen supply. And here you can see what they treat, like they treat the agrochemicals. Suppose our our land contaminated with the fertilizer. So we use and when the rainy season happens and they wash the whole soil, they wash our field, and the whole water goes to the some river body. And when it goes to river body, they make soluble to the chemicals, and they contaminate the whole river. So as per our study, what we have done in our laboratory scale, we make artificial filled in the lab in a tray, and after getting this contaminated water, we passing through this waste water to the biochar column to treat them, and we absorb these agrochemicals in the our column. Similarly, we can do for the antibiotics. I have done this for agrochemicals, antibiotics, some pharmaceutical industrial waste, some pesticide, and some organic pollutants like volatile organic compounds, which having the hydrogen sulfide and some toluene, some benzene. So what happened when we go to the anaerobic digestion process? If students are aware with the anaerobic digestion, where there is some anaerobic bacteria which act uh, and they make some fermentation kind of things, and they produce the carbon dioxide, methane as a biogas. And there in this process, there is slightly hydrogen sulfide form. And this hydrogen sulfide, after forming, it decrease the pH. And anaerobic digestion is a most important thing. It happens at around 8 plus minus uh, 0.5 8 plus minus 0.5 alkalinity. And so when H2S form, it decrease the pH and it goes to the acidic level, like 7, below the 7. And after getting the 7 pH, anaerobic bacteria stop the working and biogas production stop forming. So that's why we pass through this whole the gas to the one biochar column. I will show in the next slide like how it happened. And then after passing this biochar column, hydrogen sulfide get absorbed in this whole biochar column, and again the biogas production increased. So see here you can see the like we have studied like antibacterial study. I told like we have made the Chinese seed plants, and these bacteria were there like E. coli, plus Pseudomonas serratia, and Bacillus subtilis. And we make some microbiological student may be aware with the like a minimal inhibitory concentration, so where the bacteria will get where the bacterial growth will be stopped. This called the like minimum inhibitory concentration. So we have make some colony farming unit of the 10 to the power 9 degree, and we have checked this at OD1 at the 6 nanometer, and we fixed these whole bacteria in this experiment. So you can see here is the experiment, like we have used these Petri plates here, and we make you see these discs are the biochar dipped disc where we have fixed them inside the plates, and then we spread the bacteria. So you see here is the growth. So yeah, so see here is the bacterial growth when we use the like biochar. So what happened? Bio oil. So what happens? See this black zone is showing that there is no bacterial growth form here, and this is for the Serratia species. And see, you can see the Pseudomonas having the bigger well as compared to the Serratia, and Bacillus subtilis has this zone, and E. coli has this zone. So in figure, it is difficult to find. So when we measure and we plot the graph, we found that here is the graph, and we found that the Pseudomonas fluorescens was the showing maximum zone of inhibition, and Serratia species got the less zone of inhibition. Their growth was very less. So this were happened using the Chinese seed from the BHU campus. We have made this bacteria and this biochar and bio oil. And here is the result. Like you can see, like we have extracted the Chinese fan palm seed oil, and we have implemented on the E. coli, Bacillus fluorescens, and then you see here the maximum zone of inhibition found around 3.3 centimeter for the Pseudomonas fluorescens, and the E. coli for the 2.9. The less inhibition was shown by the 2.15 for the Serratia species, as we saw in the this figure and this graph. Another things here, I told you earlier that it has several kinds of pharmaceutically important or any important kind of chemicals. So when we compare these bio oils with the GCMS, so we found that there are several kinds of pharmaceutically important component. Like see these bio oils shown the various components like benzothiol, 2-butanamide, 4-octane 3 ethyl 7 hydroxy, and 2,3-trimethyl 4 ethyl vinyl phenol. These all the compounds are the pharmaceutically important compounds, and we have found as per the GCMS library, if students use the GCMS, so GCMS is a very good and is one of the advanced quality of the gas chromatography, which has the more than 1 lakh chemical details with their structure and their mass. And when we inject these samples, so these all the samples get compared with this library, and they tell that what the exact chemical are there. So we have found around 40 pharmaceutically important compounds, where I have given these 4 or 5 names here by comparing these researchers which have reported these things. So see, one another microbial assay study was done using the hemp bio oil. Suppose we have done this here, the Chinese synth farm, and one study has been done from the hemp bio oil, which we say the sunflower. So sunflower bio oil, and then they give the 96 whale microplates, and they used for the 5 microliter for the bacterial suspensions, and they found this growth. And this happened, yeah. I told you that this paper we have worked in US, and this paper also it's very good paper, and it having around the impact factor of this journal is around 10. So see, the title of this work is removal of hydrogen sulfide generated during the anaerobic treatment of sulfate-laden wastewater using biochar. As I told that, see, here is the column. This is the column where we have used the biochar packing. You see this black part is the biochar column, and here is the this part is the anaerobic digestion. So we made the synthetic hydrogen sulfide using K2SO4 and 1 gram of the glucose part. So what happened? The glucose was consumed by the anaerobic bacteria, and it was converted into the carbon dioxide and methane. As we know, carbon dioxide and methane is the biogas, and K2SO4. When we add the K2SO4, it started the slightly formation of the hydrogen sulfide, and this hydrogen sulfide, when it forms, the production of biogas get reduced. And here you see there this blue color ball is the storage of the gas. So when the H2S started forming, the biogas production got reduced in huge amount. And when this gas we have circulated through this biochar column, you see here, and after getting absorbed the H2S, suddenly it increased the biogas production, which show that how the biochar is helping to the enhancement of the biogas production by absorbing the hydrogen sulfide in their body. And after this absorbing this hydrogen sulfide in their inside, these biochar were applied for the crop production, for the leguminous plant. As we know that when we use the leguminous plant, the leguminous plant has the sulfur content some amino acid like cysteine and cysteine. So when we give this sulfur containing sulfur treated biochar to the field, and when it will go to the when it goes to the uh, leguminous plant, they have the good health for the cysteine and cysteine amino acids, and we are getting the this sulfur containing amino acid in our pulses, and we eat. So this was the very important study, and it was showing that how the biomass we can use as a biochar, and we can use them as a like adsorbent. If you see the AquaGuard in your home, so AquaGuard has the same things. But what happened? They are using the activated carbon. They are using the activated carbon. And when you are making the activated carbon, it is getting the more than 1,000-degree temperature to make them. Here in a pyrolysis, it is getting around 300, 400 degree temperature. And we are saving the 700, around 700 degree centigrade temperature to make them, and the application is almost similar. Okay, so we can use this biochar as a activated carbon to treat the contaminated water event. I show you that this biochar here we have used as a air pollution treatment. And here is the brief, see, and we have given here examples like, see, we have used the 200 to 2000 milligram sulfate per liter, and we found around the C here. Our biochar was able to remove 98% of the hydrogen sulfide in the concentration of 105 to 1020 ppmb, especially at a high moisture content in the 80 to 85%. And the very high hydrogen sulfide absorption capacity was found 273 plus minus 1.9 milligram hydrogen sulfide of the biochar expected to enrich the things. So see here how in research it is showing or not, it happening or not, we see here. So when we use this biochar for the same analysis, so here you see we have like make the circle, and you see the sulfur content, and here this is the XRD graph, where also this graph also showing the it is like a sulfur content graph. And here this is the image of this process, and here this is the schematic diagram of the whole process. Another example I told, like how the soil microbial metabolism happens when we use the different kind of this biochar. So see here, when you use the like ray grass green manure and biochar mix in the soil, so they are providing the some different kinds of soil nutrient, and they are making the here, see here the root, and it is showing like how the microbiome mixed together, and how they converted the elements to the plants. Like we have, like suppose we have nitrogen in the environment, and how this nitrogen goes to the plant using these microbes, and they converted them in the nitrate, nitrite, and then the root take, and then it will go into the plant, and then it made the some amino acids, uh, and then our then these amino acids go, and they store in the several kinds of product like fruit, somewhere in stem, somewhere in leaf, and somewhere in shape like sugar cane. When you go, you have like food in sugar cane, some fruits having like when you go the like potato, you can find in the root. So these things go and they make them. Another example, let's see, this is the another one for the biochar organic fertilizer. So we can use any amount of biomass to make the biochar, and we can add in the soil. So this is a very important thing. Let's see, people are saying that why we are using biochar in the land side. So in India, we have a plain and we have the desert area like Rajasthan. So when we go into the Rajasthan side, there is a less soil in not all the part, in somewhere in the desert area, and these biochar we can add them the fertilizer, and we can mix there in the desert region, and we can produce the and we can uh, crop the several kinds of plant. For example, in Rajasthan part, they are growing the watermelon. And for watermelon growing, what they do the, I show, yeah. So for the watermelon cultivation, they making the some kind of, they dig and they remove the soil and they fix the polythene inside the land, and then again they refill the things and with adding the biochar with fertilizer, and then they sown the seeds, and they are getting the huge very big quantity, very big size of the watermelon. And this technology are used in the Israel. So Rajasthan government has the collaboration with the Israel government to make sure that how the desert land we can use as a organic farming. So this is one example, and there are like several examples for the biochar. So see here is one another for the like how the oxidation process help, and biochar act as a catalyst. Suppose if there is one any pollutant, and biochar has some surface area and they have one bonding site. So when we do the some bioengineered kind of things, like when we add the some ligandic compound, like some ferric compound, for example, the ferric has a uh, three valency side. So one valency will react with the biochar, so one part, and even though it has a two valency vacant, and the two pollutant can attach there. So the and engineered biochar are also helping to get the environmental friendly to make the maximum pollutant absorption. This is also one of the very good paper published in the Science of the Total Environment, and it was done by my students, and they have removed the like when you add the ozone, it has like several bonding sites, and the ozone is a very highly oxidizing agent, so it uh, uh, deplete the big component. Like suppose we have any agrochemicals like insecticide, pesticide, which are the bigger molecule. So when we connect them with the ozonized biochar, so it make the oxidize and they break them the simple form, and then in the end it has the CO2 and the water. So see, this is the pictorial diagram of this work, like how we can use these biochar, and we can fix them like they persistent free radical, redox properties, oxygenated functional groups, and some photocatalytic systems, and some sonocatalytic system they have done, and the output was like it was like eco-friendly, economical, sustainable. As we told, like when we are using the this waste bioresource, and we are making the biochar and we are applying. So this called the sustainable techniques. And economic, when we uh, go for the chemical treatment or any other treatment, it will be costly. But here we are absorbing these things. So this is the economic process. This is the another example of the engineered biochar, how the engineered biochar remove the phosphate containing water and they clean the water. So see here there was one lake in the China. They it had the like green. So nowadays when our students, yeah, they are going to the any big city by the train or by the car. So when you enter in the city, so you see the any lake or any pond, and it has a huge amount of water hyacinth, and these water hyacinth having a nutrient content, and accordingly this study was happened because there was a phosphate rich lake, and there was these green kinds of uh, plant bodies were grown, and for, to collect them, and we made the biochar here. And after making the biochar, the water was cleaned because of the removal of these things, and then these treated biochar were used here to the crop production. Because when we make the pyrolysis process, so this biochar having the huge amount of the phosphate, and phosphate is the one of the fertilizer source, as we know. So here is the crop production increased. This paper was also published in a good paper. And nowadays, people are asking like artificial intelligence, machine learning. So we have aligned our work with how the machine learning will help to the our biochar application. So suppose if the biochar has treated, so we can calculate the nutrient recovery. We can do the some color removal study, some electrochemical sensors we have used by the machine learning process, and the like AI and ML the present demand, how the carbon dioxide absorption happens. So we can propose this study with the help of the AI and ML things, and then accordingly we can work in our laboratory scale, and then we can make the data artificial neural network and some reverse uh, study in this whole process. So see here is the like how the ML machine learning and artificial intelligence help in the biochar study. So here it is showing like how the biochar is a promising carbon sink process which absorb the pollutant and to clean the wastewater, waste air, and the several things. And so after going to this, so nowadays we know like United Nations has fixed the some sustainable development goals, and these goals are the 17 goals, and nowadays everyone has to align their work with the uh, these sustainable development goals. So we have aligned our this work air pollutant removal, wastewater treatment, and we aligned that how our work is aligned with the few sustainable development goals. Like see, the one is like showing like how the good health and well-being happens, and then the quality education. So uh, the justification is like suppose if there is a water contamination happens, and when we and in India, the maximum disease happens because of the contaminated water, like dysentery, cholera, and several things. So if we use this bioresource to biochar to treat this uh, water pollutant and the air, so we will get the good health. Our health will be okay. So this is the third goal of the United Nations Sustainability Development Goal, which is showing that how it will help. Then the quality education. So how it will help to the quality education? So suppose when you do some treatment system for the wastewater and some air treatment system, so there will be industrial part and there will be the poor people will work there. And when they will work there, they will get some money uh, from the job, and then they will give to the quality education to their kids. And the sixth is the clean water and sanitation. So similarly, if the when the water will be like clean, so we can provide the clean water as per the 6 number sustainable development goal, and there will be the like hygienic things will be there. And the ninth one is the industry, innovation, and infrastructure. So the treatment of wastewater and the treatment of waste air is a very huge industry. When you go to the any wastewater treatment plants like sewage treatment plants or household wastewater treatment plants, and another one is the like industrial wastewater treatment plant. So when you go, it is a very big industry. It is a very big industry, and it provides thousands people in a one industry. It provides more than 100 people for the only wastewater or waste air industry. And there is a so many chance. There is so many things to get the innovate. And suppose when you treat the wastewater and then you package the water for 200 ml to 500 ml in the packaging, it is a kind of innovation. And then after this treatment, there is just settling of these pollutants, some solid party there, and these pollutants are work and they act as a biofertilizer. So we can use them as a small scale fertilizer industry, biofertilizer industry. And after that, obviously when the industry and innovation will come, then infrastructure will be the developed. And one is the like sustainable cities and communities. Like there is one village in Sikkim, which are the completely sustainable village. So accordingly, in our Indian cities, we can do the sustainable city, like all the product from the cities, and they can use in city and they uh, finish. Like, for example, in Indian village system, our village system was initially it was the sustainable village. How it was sustainable? Suppose there was no plastic. So when we use the fertilizer using the cow dung or any animal dung, it goes to the land, it goes to the our field, it acts as a fertilizer, and then we grow the crop, then we cut the crop, human being use the cereals and other things, and the fodder goes to the animals. So this is the circular economy, and everything by the sustainable. So they produce the product in the village and they finish in the village. So this is why the like sustainable village and sustainable cities and their benefit to the communities. And these goals we cannot achieve without working in the partnership, because suppose for these work we need the like for example we need the money. So when we go for the any small scale industry makeup, so we need the some fund. So this fund will given by the some government industry or some industry partner. So then there is a need to the partnership for to achieving these goals. So this is the few SDG which I have aligned. If we aligned this study, it will be like more SDG could be aligned, but I made around 5, 6 only. And this is like on the basis of these things, I have my research publication. These things I have published around 150 plus publications with 7,600 around Google Scholar citation, and these are the things. And as I told to ma'am that my presentation will be in the second part. So I am going to share like nowadays these studies are more important when we add the startup culture. So see here using these things, where using these bio. So I want to show one another project. So is my slide visible? This Biojagat Private Limited. Anyone can
Yes,
tell?
sir. Yes, sir. It's
Yes,
visible,
sir.
sir.
Okay.
Visual.
So see, ma'am, so this the earlier presentation why the some academic kind of things and some like uh, for students to get the academic PhD and masters, and then nowadays the most important things is the startup culture. Government is giving a huge amount. If anyone has any idea to make the like startup, so government is giving money to nurture them. So using this kind of experience, I have opened one startup, and I use these agro products using the bioresource, and I grow the mushroom, and using these mushrooms we make the cookies, mushroom chunks, some nursery pots, and several kinds of things. And I have registered this as a Biojagat Private Limited, and I am proudly saying that my this startup has been accepted for the financial support from the government of Uttar Pradesh, around 20 lakh rupees. And here in my university they have also selected for them. So see, so how this idea came to us. So when I was working in the IIT Guwahati, so it was proposed that why we are not using the startup culture to open the industry. So as I'm the microbiology student, and I thought when we use the biomass to grow the mushrooms, and then we can use these mushrooms to the value-added products. Like suppose there are so many mushrooms which are the medicinally important mushrooms, like shiitake mushroom and some other mushroom, and some food important mushrooms. I'm not talking about the some kind of button mushroom and oyster mushroom. These button and oyster are the very common mushroom, which are the very less amount mushroom. But for the learning process, students can learn from these oyster and button mushroom, and but when they get experience, then they can shift to the high yielding mushroom, like medicinally important, good quality of food mushrooms. Like when I was in Taiwan last year, I visited. I saw that they have several kinds of mushroom items in their restaurant. So they were giving the mushroom soup to see, to drink, and they have the several kind of mushroom dishes. So, and we know that mushroom cultivation it requires very less land. It is not needed the that much land. It is like a vertical cropping. So see this here. Here is the my startup things. It is in IIT Guwahati campus, and here is the some products. Like this is the some biscuits. Here is some pickles. Here is some cake. Here is some chocolate, some candy. This is this is up to the startup scale, and now it has selected for the financial support. And then that see the like Kalasvi and who is the secretary of the CSIR and Ministry of Science and Technology, and he is Professor Bimal Katiyar, who was my mentor, and he the person Dr. Bipin Chand Shukla from the DSIR, Department of Science and Industrial Research. So they are nurturing these startup to support by finance. If you have any idea, so for nurturing your idea, they give 2 lakh, 3 lakh rupees to as a seed grant. And when it will go to the like implement, then you can get the 5 lakh, and then they have several kind of steps to get the more money. So I have signed the MOU with the IIT Guwahati in the presence of these big guys, and they are the witness. One is the director CDRI, one is the he is the director ITRC. Behind me, this is the director NBRI in the Lucknow. So four directors were there in the this IITR campus, and it happened under the umbrella leadership of the CRTDH New Delhi. And this is the certificate of incorporation of my mushroom cultivation. And here is the some proof of concept. So see, I have given even the topic like parali biomass as an agro waste, and the Himalayan biomass cultivation into the mushroom and its residual utilization as a biopesticide and biofertilizer to make the value-added products. And we have huge amount of and this whole process is the uh, livelihood creation, waste to wealth, circular bioeconomy. So see, uh, it happens with the mushroom cultivation, and it happens mushroom grow in the some kind of hut. So we have given the this project name is the Muskurahat. So Muskurahat is the mushroom, and hut is the the some kind of a uh, vegetable, some kind of a biomass home. We can, so we given the Muskurahat term, and Muskurahat means the smile face. So we have given our idea name is the Muskurahat. And these all are the products of the biochar and this biosource utilization and several things. And these are some expected outcome. We can give the training. It will help to the Atmanirbhar Bharat. Female farmers are getting benefit using these things. The government of India has one Ladli scheme where they give the 2 lakh to the female farmers to get the Lakpati Didi scheme. So if they will do in their home, they can easily get the, at least they can make themselves as a Atmanirbhar. They can make them a entrepreneur. Okay. And these whole things are like how these things are connected with the aligned with the sustainable development goals. So this all the things biochar muskurat is the come helping to the maximum 17 goals of the this United Nations. Yeah, and this is the things. And so I have claimed around 46 lakh. They are giving around 20 lakhs rupees. And similarly, you can see here we have made the. So this is my this is the company's website. You can see the Biojagat is the mushroom ka daur, and here is the see the address. It is in IIT Guwahati, and here is the some old logo and these things. Here is the, you can be the CEO of your company. You can be your director of the company as the entrepreneur, and then she is my wife. She is also one director in the company, and these the different kinds of mushroom which we grow in the IIT Guwahati campus. So see here is the some chantreli mushroom, shiitake mushroom, which is a very benefit. It is people are selling this as a 4 lakh rupees per kg. This is a mural mushroom. And this I told you, like this is a vertical cropping. So he is my son. See how he has taken the product up as a mushroom. So my whole family is in this business. Even though we are in the startup phase, but we are in. And see, this is the like a vertical cropping. See, so you need the very less place. You need the, see, this is the box, na. So you need the very less space to make the vertical cropping, and you can get the maximum product. And then you can go for the several kinds of value-added product. Like I told, I shown the figures like uh, mushroom biscuits, mushroom chunks, some mushroom pickles, some mushroom nutraceutical powders, several. There is an endless product if you want to work on this. So I'm just, I have given one example for this startup. Even you have any idea, you can also open, you can, and I can help you to get the, how to get the like financial support. I can guide you. I can guide you how to open the, this startup culture, because many people are uh, getting connected with me. So after this, I want to close the my talk, and in the end of, I thanks to madam. Ma'am is my senior. When I was in IIT, when I was in Niri, so madam was my senior, and she, so I, I went from a very village place, and in Nagpur, madam department was EIRA, and she taught me a lot of things like how to get the good education, how to learn the things. And I'm proudly saying that, ma'am, since 2007, you have connected me. You have not forget me. And
Thank
kindly
you so
bless
much.
me. Yeah,
Thank you so
kindly
much.
bless me. Yeah. So
Sir,
it is almost 20 years to we are in connection. And when I entered in the NIRI, ma'am were like found, and my academics as a researcher was zero. Nowadays, I am in top 2%. Many things I have achieved, roamed many countries, and because of the seniors like them. Otherwise, it will be difficult. If someone will show the wrong path, you will be killed. But if someone will keep you on the right track, you will be going the right path. For example, if someone will tell you to sit in the passenger train, so you will be go very slow speed. And if someone will put you in the Duranto Express, you will go immediately in the Delhi. So that's why you need the seniors like Devanshi madam, who were the like very energetic people. And thank you, ma'am. Thank you. You you never forget me. Yeah.
sir, I would like to thank you personally that the privilege is always mine to get in touch with you. It's always good to see you at a very good place. I'm always getting inspired by people like you. In fact, I would like to thank you that you have really brought a lot of energy into the participants as well. They have really gained a lot of insights and they are really motivated to get into the startup culture. So I open the dais to the participants. Please unmute your mics and get in touch and get interactive with Balendu sir. You have got a good opportunity.
Yeah. So I request the participant for the further contact. You can take the this picture, so you can get the immediately response within half an hour. And yeah, please ask if you have any doubt, if you have any things.
Dr. Balendu, good evening. This is Dr. P.K. Shukla from Prayagraj. Your lecture was very, very interesting. And
Okay, namaste, sir.
I have one query and one question that I have designed one project regarding with the application of biochar for treating sewage wastewater. So my question is, if I am using that biochar, then which property of the biochar I should consider the most? I mean, the absorptive property, ion exchange property, coagulative property. Which property I should consider the most?
So, sir, for the wastewater treatment, we consider the mainly the structural property of the biochar, sir.
Okay, okay.
And sir, if you sir make some kind of bioengineered or engineered biochar, like if you made some ligand treatment, so
Mhm.
it creates the maximum bond site to get the maximum pollutant in. And sir, I visited MNIT frequently twice in a year. So if you say I can
You are
visit,
most welcome, sir. I am from Schwartz. It is previously known as Allahabad Agriculture Institute deemed university.
yeah, sir, I know Sir Schwartz, I know. So I
Yes, yes,
can visit
yes.
for definitely. So maybe in
Actually,
January I will be there.
okay, okay, sir. You are most welcome, sir.
Yeah.
And I will be in touch with you. I will give my email ID. I will contact you with the email, sir.
Sure, sir, sure, sir.
And
Actually,
and
sir,
we are using the biochar from the rice husk. And
sir, sir, during my IIT BHU tenure, in
uh,
BHU, it is like a huge bioresource, sir.
okay,
So we have like rice husk biomass. We have in
I
campus BHU, there is just several kinds of like some weeds kind of things, some Chinese seed farm. We have even with that mango kernel, sir. जो आम की
mm,
कुटी होती है, sir, we have made this as a biochar, and
okay, okay, okay.
I have worked with Professor.
We are, sir, having the two type of biochar. One is a rice husk, other is a water hyacinth, Icornia. Jal
Yes,
Kumbhi
sir,
जिसको
water
बोलते हैं.
hyacinth. Yeah, water hyacinths are very
So that
important.
is a very good absorptive, especially heavy metal absorptive property.
Yes, 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 sir. Yes, sir.
Uh,
It is
and
very good for the chromium, copper things.
yeah, yeah. When you will come, sir, we will make you visit our plant
Okay.
tissue culture lab.
So, sir, I request you just you send one hi to this number. Both are the WhatsApp number.
Okay, okay, sir. I will do that. I will do that. And we
And
have a very good
I'm
plant
also
tissue
from
culture
the
lab.
Uttar Pradesh, Balia. So it will be always good to.
So you are Bagi Balia, yeah, right. So.
Yes, sir. Thank you, sir. Thank you.
Okay, sir. Thank you, sir. I will be contacting with you, sir.
Sure, sir. Sure.
Thank
sir.
you so much.
Any other participant who would like to have a talk with sir, please unmute yourself.
So see you guys, I always tell that if a person like me can reach to this place, anyone can reach that place because of your hard work and in a disciplinary way.
Thank you so much, sir, for giving a wonderful expert lecture. And it's really nice to see you at such a wonderful place. And I'm hoping to see you in person somewhere in future again. And you've really inspired us by your startup. And we'll get in touch with you back again, hopefully with some new project also.
Sure, man. Sure.
Thank you so much, sir.
Thank you,
And
ma'am.
thank you from IPS Academy Institute of Engineering and Science. On behalf of Civil Engineering Department, I'm really thankful that you've given your experience shared with us and also inspired us today. Thank you, sir. Have a good day.
Thank you, ma'am.
Thank you, sir. Thank you, sir.
Please be connected, my dear students.
Thank you to the participants for being active, and we'll see you tomorrow at 6 p.m. in the evening again.